Well, good afternoon, good evening, good morning, wherever you are joining us for this CDDF webinar. My name is John Smythe. I have the pleasure of moderating this session on measurable residual disease in acute myeloid leukemia. As many of you will know, the CDDF is a non-profit organization. We're registered in Belgium, and we provide a neutral platform to stimulate interaction between all the stakeholders involved in cancer drug development, as illustrated on that slide. I have the privilege of being chairman of the board at the present time, and this session is going to be co-moderated with my colleague Axel Glassmarker, who will join us in the discussion session. The next slide, please, Richard. Just a few technical points. Please report any technical problems via the chat function. The questions, which are very important, we really want your contribution. These can be asked at any time via the Q&A function. We will present your questions to Christopher Harrigan during the discussion session. And we find that if attendees can double click on the Zoom screen to enter or exit the full screen mode, you'll get better visuality. So I've introduced Axel and myself. I'd like very much to introduce Christopher Harrigan. We're very fortunate indeed to have, if I can have the next slide, please. We're very fortunate indeed to have Chris Harrigan joining us for this session. He's had an extraordinarily distinguished career, which started very sensibly in the United Kingdom, where he trained in Oxford. He subsequently worked at Johns Hopkins, but he's been now for some time at NIH as chief of the Laboratory of Myeloid Malignancies. And he has a very distinguished CV and publication record, specifically recently focusing on this important issue of what used to be called minimal residual disease, and we now, since about 2018, we've called measurable residual disease. We know from previous workshops how important this subject is to people that join us at CDDF, and in some ways this webinar is an hors d'oeuvre for a workshop we're going to hold in hybrid fashion in Amsterdam, if you can join us, on the 25th and 26th of April. But we're very keen indeed to get into the subject now with a world expert. So Chris, the stage is yours.
Hello, my name is Christopher Harrigan. I'm Chief of the Laboratory of Myeloid Malignancies at the National Heart, Lung and Blood Institute of the National Institutes of Health. I'd like to thank the Cancer Drug Development Forum for this opportunity to talk to you today about MRD by NGS in patients with AML undergoing allo transplantation. I do have some disclosures. I run a laboratory at the NIH, which has received funding from Cellus, and we have active research collaborations with QIAGEN, Archer, TwinStrand, and Mission Bio. I've served on AML MRD advisory boards for the pharmaceutical industry as part of my official duties without compensation, and I work clinically as a leukemia physician at Johns Hopkins. As an NIH employee, I work for the US government. I do not speak for the US government. And today I have 30 minutes, so I just want to talk to you about two things: where we are with AML MRD, and where do we go next? So, where are we? For this first question, I'll break it into three parts. What is AML MRD? What's the evidence that AML MRD is prognostic? And can we as physicians do anything about it? So to give you some background, over 20,000 Americans every year are diagnosed with a highly fatal blood cancer, acute myeloid leukemia, AML. While scientific understanding of the etiology has improved considerably over the past decade, with 9 new FDA drug approvals in the past 5 years, the depressing reality is, while we have the ability to achieve an initial complete remission in many, what I'm showing you here is a cartoon representation of that in 3 patients, with time on the x-axis and leukemic cell burden on the y-axis. These remissions, however, are not maintained, and death from leukemia remains the most likely outcome. Relapse has been described as the most important problem in AML. And I think that's completely wrong. We artificially dichotomize leukemic disease burden, which at first approximation is a continuous variable, into remission or relapse on the basis of grossly insufficient tools. So what are those tools by which we measure the success of therapy? It turns out that the criteria for determining a remission in AML changed little for 60 years, based on cytomorphological assessment and a complete blood count. But as far back as 1980, there was this understanding that a clinical complete remission, when measured this way, was a highly heterogeneous state with clinically diverse outcomes. There was often nothing complete about a complete remission when measured with these low sensitivity tests. So going back to our apparent relapses, we know if we use higher and higher sensitivity tools, we can get a better understanding of residual disease burden. Now, suddenly, our 3 complete remission or CR patients don't look the same, and it isn't so surprising they had different outcomes. We see that as we use higher sensitivity tests to quantify residual disease burden, we get a better appreciation of the impending clinical outcomes. We call this measurable residual disease, or MRD. So if we have known about MRD and AML for decades, why hasn't it been better integrated into clinical care and drug development? Well, we know that AML is just one name used to describe many diseases, whether that's at the level of cytogenetics, the combination of somatic mutations that can go together to make an AML, or even the multiple subclones that can be present within one patient. However, in response to this, the ELN did update the response criteria in 2017 to include a higher stringency best response category of MRD negativity. And we've now published 2 ELN consensus standard of care guidelines for the use of AML MRD, most recently just a few months ago, shown here. Essentially recommending the use of molecular methods for those when you have a pathognomonic marker to track, for example, in APL for core binding factor leukemia, for mutated NPM1, and in the rare cases of BCR-ABL, and flow cytometry for everyone else. So what's the evidence is prognostic? AML is a disseminated disease at diagnosis. By analogy to solid tumors, it's always stage 4. And while we can often achieve a complete remission, as I've shown, those remissions are illusions. And often the disease becomes clinically detectable again in the first couple of years after treatment. For those at higher risk of what we call relapse, we therefore offer additional consolidative therapy to those who can tolerate it in the form of an allogeneic hematopoietic cell transplant, the original cellular immunotherapy for cancer. And while this is effective in reducing relapse in most AML subtypes, the most common mechanism of treatment failure, as you can see here in the blue, is still relapse or refractory disease. We know that people who enter allo transplant in remission do better than those who enter with active disease. The outcomes for those entering in complete remission can still vary considerably, however. And MRD testing while in remission prior to transplant can explain much of that heterogeneity in better risk stratified patients. What I'm showing you here are 2 independent studies on AML patients that are a key landmark of clinical remission prior to an allo transplant. On your right is our study from the NIH, measuring MRD using molecular methods on blood samples. On the left is a much larger study from the Fred Hutch using flow cytometry on bone marrow. Shown in yellow are the poor survival outcomes for those who entered transplant while not in remission. However, in gray, we show that those who are in remission but have MRD detectable had similarly poor outcomes. So despite the large methodological and cohort differences between these 2 studies, the central finding is the same. Measurable residual disease immediately before a transplant is associated with poor clinical outcomes after a transplant. Here's a meta-analysis of many studies from AML patients, all at that same pre-transplant landmark, all showing the same effect, that regardless of how you find it, residual disease before a transplant is associated with worse survival after a transplant. And that association goes beyond that particular treatment time point, however, and actually holds throughout treatment, as shown here in this more recent analysis of 81 studies and over 11,000 patients. So, can we do anything about it? I've shown you the 2 meta-analyses, both demonstrating that in AML patients in remission, but with measurable residual disease, have high relapse and worse overall survival. What was not known is that MRD was just useful in determining prognosis, but can we as doctors do something about it? A retrospective multi-center analysis from the EBMT has suggested that younger patients undergoing transplant in remission, but with MRD, benefited from treatment intensification. But to answer this question fully, we needed a large randomized clinical trial. One scalable intervention is the amount of chemotherapy conditioning given prior to a transplant. In 2011, the Blood and Marrow Transplant Clinical Trials Network opened a phase 3 randomized control trial to compare reduced intensity conditioning RIC versus myeloablation MAC regimens in myelomalignancy patients under the age of 65 in remission, with the primary outcome being 18-month overall survival. And as you may know, this trial was stopped early after just 76% of planned patients enrolled due to the high relapse rates shown here in those randomized to reduced intensity transplant. Of 228 AML patients in CR randomized to MAC or RIC, we were able to get hold of pre-transplant remission blood samples from 190 of these. These were flash frozen blood samples. The ELN approved assays at that time, quantitative PCR or flow, required RNA or intact cells, so it couldn't be done here. We could extract DNA though. The issue is we had no information on what mutations to look for in that DNA. This is what made this the hardest problem we've worked on to date. We had to find a way to do de novo variant calling at 0.1% or less allelic frequency, 50 times more sensitive than usual NGS variant calling you may use in the clinic. Next generation has multiple sources of introduced errors, resulting in false positive error rates when considering allele frequencies less than 2%. We overcame this with a combination of laboratory and bioinformatic approaches to make clustered consensus variant calls. And then added on a background error rate model to maximize specificity. Our hypothesis here was simple. It'd be those in clinical remission but with detectable residual leukemia using high sensitivity testing who would be harmed by reducing the intensity of chemotherapy. We found that the prognostic implications of the detected variant varied by the variant class. On the left, I'm showing you the two most extreme examples, but also varied, as I'm showing on the right, by conditioning intensity. For patients who tested negative, it was about 60%, survival was the same regardless of conditioning intensity. For those testing positive prior to transplant though, we saw higher rates of relapse, a 3-year cumulative incidence of 72% versus 15%, and lower survival rates, 3-year overall survival 34% versus 59%, when randomized to RIC rather than MAC. So this study provides the strongest evidence to date that intervention for AML patients with genomic evidence of MRD can result in improved survival. The National Cancer Institute, the NCI, together with the cooperative groups ECOG, SWOG, the Alliance, the Canadian Cancer Trials Group, is now launching an international master protocol umbrella trial in precision medicine for AML that will use AML MRD testing as an endpoint, as an inclusion criteria for subsequent iterative tiers of therapy. Using flow cytometry and NGS techniques such as highly sensitive duplex DNA sequencing, assays will help answer key unanswered questions regarding AML MRD. So what's next? Again, I'll break this into 3 parts. So AML MRD testing can be used in many ways. As patient selection or eligibility criteria, as I've just described in the MyeloMatch trial, to assign high and low risk cohorts based on a threshold level. It can be used for monitoring post-treatment of individual patients, typically with serial measurements, looking for patients who turn from negative testing to positive testing for early detection of relapse. And it can also be used in drug development to get a bright signal of anti-leukemic efficacy by looking for the depth of remission by monitoring specific targets. What I haven't mentioned here is the idea of surrogacy as a regulatory endpoint, showing for a regulatory endpoint such as overall survival, which is beyond the scope of this talk. So we have some known knowns. AML patients in a CR have a higher risk of relapse if evidence of residual disease, regardless of how you test it, although NGS is still an evolving field, is detected prior to transplant. So MRD test positivity is prognostic. Patients are underserved by the current standard of care, and should be offered a clinical trial. MRD test negativity, however, does not equal patient MRD negativity. Patients testing MRD negative by flow prior to an allotransplant can still relapse, about 20 or 30% relapse risk. NGS testing for MRD currently has a high rate of false positivity for relapse. Almost every patient will have some kind of mutation detectable in remission on a broad and deep NGS test. And the concordance of NGS with flow and the clinical utility of MRD testing will have to be validated for specific clinical scenarios and prospective clinical trials. For example, the Myeloma randomized phase 2 trials as I've described. And then some patients in CR prior to an allotransplant will have been incompletely treated because the criteria for success of CR was insufficiently stringent. They may have chemosensitive disease left to treat. In this way, MRD may be a predictive biomarker. For example, myeloablation versus diffuse intensity in the 0901 study. Again, MRD test status reflects only the sample that was tested, not the patient. False positives and negatives on a single test are to be expected. Kinetics and serial measurements may be superior to single landmark assessments. And then MRD itself may give us information on the residual chemoresistant clone, which may or may not be resistant to novel agents, and this is testable. So secondly, I want to take you through the data in this paper to give you a sense of what the current state of play is for clinical testing in use. This isn't our work, but it's informative as it reports AML MRD testing of very high quality in consecutive AML patients treated with allotransplant at a single center. So this is from the Fred Hutchinson. The age of people transplanted was 18 to 80 with a median age of 56. Most were in CR1, 77% were in CR1. And the duration of CR prior to transplant was a median 96 days. This isn't a randomized study. It makes no sense to use it to try and understand the impact of condition intensity, given the huge bias of physician preference at a single center. But what makes this study interesting is its gold standard MRD testing by flow cytometry on consecutive patients, as described here. So out of the 743 transplants, 31% relapsed. And 147 patients have tested positive for MRD prior to transplant, or 20%. When we look at the connection between those two factors, though, there's a disconnect in terms of the sensitivity of this testing to predict the relapse event. Only 96 of the 230 relapses were predicted by pre-transplant MRD. And of those patients who tested positive, only 65% relapsed. So if taking all those who relapsed, more of those are tested MRD negative prior to transplant than are tested positive. So despite the fact that MRD testing prior to transplant is a very powerful risk stratification tool, in terms of the sensitivity to detect a relapse post-transplant, the sensitivity is suboptimal. There's many potential reasons for this. It can be the wrong time point. It's a single landmark. Potentially, you can imagine serial measurements post-transplant help. Maybe the wrong test. The MRD is there, but not recognized as AML. Maybe insufficient limited detection. The MRD is there, but the sample is incompletely characterized. Maybe the wrong sample. The MRD is in the patient, but not in the tube. And then it may just be the wrong premise. Who said that MRD testing will detect 100% of relapses? But this is, this gives you a sense of the current gold standard in use. And then finally, some of the known unknowns. And these are technical aspects, but the degree of discordance between MRD measured by different techniques is still an evolving area, and I'll take you through some data on this. MRD measured by flow cytometry, which is the current ELN approved method for those who don't have a mutation that can be tracked by qPCR, may be discordant for that detected by NGS. And those edge cases are an area of active research. It's unknown if blood can substitute for marrow in flow or NGS MRD measurements. There's obviously considerable clinical benefit if that could be the case. And there's some data from quantitative PCR testing of NPM1 that blood may actually be a superior way to track disease burden by MRD. We don't know if all detected non-DNMT3A, TET2, or AXXL1 mutations are appropriate for tracking in remission, or are some more pathognomonic than others. We know that late pro-proliferative mutations, the FLT3 ITDs, the RASes, can come and go over time. And alternatively, we know some very early mutations in leukemogenesis may persist in a pre-leukemic clone and not necessarily portend relapse. And so working out exactly what the significance of detecting any particular mutation at any particular time point is going to take more data, and I'm going to show you some ways that's going to be generated. We don't know how often subclones are responsible for relapse. I actually found in remission. And if we do MRD depth testing of the original diagnostic sample, can we get a clue of those potential escape clones before the event? And then finally, we're increasingly moving into an era where our intensive cytotoxic therapies are being replaced in many patients by less intensive therapy. Combinations of hypomethylating agents with BCL2 inhibition, for example. The prognostic significance of MRD negativity is going to depend on the type of treatment given. And we just haven't yet generated the data sets to be able to say with high confidence what MRD positivity or negativity means in every one of those settings. To give you just one example of some of the technical challenges and the discordance between techniques, I'm going to show you here an extremely detailed investigation of the differences between flow cytometry and next generation sequencing in one of my own patients with relapsed inversion 16 AML that I treated at the NIH Clinical Center. So clinically, what I knew was there was a 5% CD34 positive population of abnormal blasts. Cytogenetics showed a 47XY karyotype in this man with an inversion 16 and a trisomy of chromosome 8 in 4 metaphases. There was a mutation in DNMT3A noted on clinical DNA sequencing. And we found additional mutations in DNMT3A and TET2 in our research sequencing. So what we did in the research lab was perform whole genome sequencing on flow sorted AML to exactly map a base pair resolution inversion 16 breakpoint, and use that information then to design a patient personalized single cell DNA sequencing panel, which we use with antibody oligoimmunophenotyping. Now, obviously, this is a research use only investigation, and we could never go into this kind of depth into every patient in practice, but really we did this to really as an exemplar of some of the ways these tests can be discordant. There's a lot of data to take you through on this slide, but I'll take you through it. And so these columns on the bottom represent individual genomic features: DNMT3A mutation, DNMT3A mutation, TET2 mutation, the trisomy 8, the inversion 16. And the rows represent the clonal populations. So leukemic, CHIP, and wild type hematopoiesis. And these are defined by the presence or absence of these features. As you can see, the AML clone is independent of these clonal hematopoiesis CHIP clones. The bottom line of this study, as predicted, is that neither NGS sequencing or flow cytometry tells us the whole story. In this case, reinforcing the previous finding of us and others regarding DNMT3A and TET2 mutations as being unreliable markers of residual AML. Here, they're completely independent from the residual AML clone. Flow cytometry in particular was relatively specific, but not sensitive, only identifying one of 2 predominant AML immunophenotypic populations. And I'd like to finish by highlighting 2 major new initiatives made public this month that should finally help us fulfill the promise of AML MRD as something that's not just good for papers, but something that's good for patients. Earlier this month, on the 8th of February, a 4-year, $6.5 million Foundation of the NIH project was announced, focusing on genetic tests to improve the accuracy of current AML MRD detection approaches. The project partners include the US Food and Drug Administration, the National Cancer Institute, 12 private sector partners, including AbbVie, Amgen, AstraZeneca, Bio-Rad, Genentech, GSK, Jazz, Novartis, Cimex, 10x, Thermo Fisher, and TwinStrand Biosciences, with leadership from myself at the NIH and colleagues from 2 academic partners at Harvard, the Dana-Farber Cancer Institute, and the Fred Hutch. We're going to work collaboratively to try and test new technologies against established methods for MRD detection and try and design better tests that can be integrated into clinical trial design and drug development, really focusing on 4 key areas. Developing reference materials that can be used as standards for benchmarking across assays. Secondly, create a process through which assays and new technologies can be developed and tested. Thirdly, validate new assays and develop a public data set where the performance characteristics of assays can be compared. And finally, collect data to support the use of MRD as a validated indicator of treatment results in clinical trials. The second major initiative, not formally announced yet, but posted on clinicaltrials.gov on February the 8th, is the MEASURE protocol. So the CIBMTR, in collaboration with the National Maradona Program, has agreed to sponsor a national level, multi-center prospective study to establish a national framework of introducing measurable residual disease into the clinical care of AML patients undergoing allogeneic transplant. By providing diagnostic material, serial monthly samples of blood after transplant, and blood and marrow samples at the time of relapse, this study will seek to optimize molecular monitoring for relapse, while also studying the biology of relapse. It's due to open in 12 high-volume US transplant centers in July of this year. Enrollments are expected to occur over a 4-year period, with up to 1,000 subjects in total. So to summarize, there's nothing complete about a complete remission in AML. But using higher sensitivity tools, AML MRD is prognostic for relapse and survival. We now have higher stringency ELN response criteria we should be aiming for, the idea of a complete remission without MRD. And the current ELN recommendation you can read in Blood from last year, but is molecular testing for those with a pathognomonic marker, flow for everyone else. Next generation sequencing is described as an adjunct for now. There's a great deal of data coming soon, and I expect the later iterations of the ELN recommendations will include the use of as NGS, this technique is likely to take a larger and larger part in AML MRD testing over time. Assays currently available, however, are not typically fit for the purpose of MRD, with some notable exceptions. And the clinical utility of AML MRD needs randomized controlled trials, and those are coming soon. I'll finish with a picture of my lab who generated some of this work. This is prior to the pandemic, when we could be in close contact. And I'm happy to take questions now, but if you don't want to take questions, if you don't want to ask me a question now, you can always contact me on the contact details shown here. Thank you very much.
Well, Chris, thank you very much indeed for an extraordinary, comprehensive discussion. And I'm glad, I'm sorry you had some technical problems earlier in the morning for you. But in a moment, obviously, I'm going to bring Axel in and ask, and we've got questions on chat. But can I start right at the beginning? Because not being an expert at all in this field, you describe extraordinary complexity within one lab or one center, and saying that some of the tests aren't ideal, and you've compared flow with NGS. But when you're getting into the field of multi-center trials, can you comment a bit on the quality assurance? I mean, if you swap samples with the Fred Hutch from the Hopkins, what sort of degree of concordance would you accept? Is that a fair question?
Absolutely. I'd say arguably it's the key question. We want to know that if you're tested in one center, there's comparability with the people who are tested in another. And I think, one of my partners on the FNIH initiative is Jerry Radich, who obviously went through this with CML to have a validated biomarker. The complexity in AML is just a level higher, obviously. I think that's a key point. We've started doing some of this with the European Leukemia Network with essentially round robin testing, where we're testing unknowns in different reference labs. But part of the both the FNIH and the measure protocol will involve both site-specific testing and centralized testing to really help benchmark some of these standards. The issue, frankly, has been that there hasn't been a push to harmonization. The incentive for academic investigators is novelty, and there hasn't been one dominant technology that's really shown to be clearly superior to others. And so we just haven't had the harmonization needed to come around one standard. But I think these new initiatives, I think the case has been made that we're not going to get anywhere by all publishing our individual papers with wonderful methods. We need to harmonize, and I think that message has come through.
I hope very much that we can tease this out when we get to our workshop in April, because you made a point. You don't want to discuss the role of drug regulation in this, but one can absolutely imagine that the FDA will want more reliable cross-center evaluations for things if they're going to be approval. Let me bring Axel in. Axel Gosmark. He's not been very well today. It's brilliant you can join us, Axel. So.
Yeah, thank you. And hi, Chris. Thank you very much for the great talk. Shall we jump directly into the discussion? The first questions are coming in, and please feel free to type your questions into the Q&A function. So the first question to you is a rather complicated one. Recent data in NPM1 AML by Andrew Wai and Tim Lay indicated that not all MRD positive patients will relapse, and that immune TME dysregulation could be critical for potentiating early relapse. Any thoughts for the future of MRD assessment, refined prognostication, in particular with regard to TME components?
Yeah, so that's a great, very expert question. That it's clearly not going to be as simple as just disease quantification. That there is a host involved, there is a patient involved, and we know even outside of the allo transplant setting there is a host immune response in remission. So both for early and late relapse, I think there's evidence that of immune control and immune escape of AML. Integrating those biomarkers into, I think, as a first pass, I think the push for the next 3 or 4 years, something from my perspective, is to have robust disease quality and quantity markers to say what disease is present in the sample we're measuring across sites. And I think really understanding which components of the immune system and the tumor microenvironment are most important in disease control is largely going to be predicated on having reliable markers that we can test at multiple centers. So my hope is we can at least lock down one of the variables, and then explore integrating immune correlates of disease control.
Yeah, thank you very much. Maybe we go just to the next question here. Is a personalized panel or a fixed panel ideal for MRD in AML? I think that should probably be answered for NGS and for flow separately.
Yeah, so there's many layers to that onion of a question, I think, right? So there's the from the patient perspective, where obviously you'd prefer a panel that was personalized not just to your mutations or immunophenotype, but specifically for the leukemic clone and the subclones that may result in relapse. There's the logistical questions of how possible that is in the current structure, and how well that can be done in real time. The example I showed you, that work probably took a year of people in the lab to really fully understand where NGS and flow are working. We need something with much faster turnaround than that. And then, the final, I think one final piece of that onion is just, or one layer of that onion is, we're wrong, and we can be wrong. We're not smarter than the leukemia. And I think we've proven this time and time again that we can target one marker, and the FDA guidance to industry was stated this very nicely, that we shouldn't just track the target of therapy. And so the predominant clone we're treating may not be the one that's responsible for relapse and death. And so I think even in a personalized panel approach, you would still want to allow for the possibility that other mutations may develop that weren't detected at initial diagnosis. And so I think my long-winded answer is why not both? I think we will have a deep validated panel that can be used across many patients that would account for some of the possibilities of evolving escape mutations in proliferative mutations, but also allow the possibility of adjuncts to that for those patients who wouldn't be covered optimally by such a panel. And you can imagine a future just as in the cell-free DNA world where tumor profiling with whole exome or ideally in AML whole genome sequencing could really inform adjuncts to a well-validated standardized panel. Depth is going to be arguably more important than breadth, though. And I think there's always this temptation to have a bigger and bigger panel of every potential drug candidate, every potential subset. And there's a long tail in AML where you may end up with looking very, very broadly, but missing the more common mutation by not having sufficient depth. And so I think it's going to be a hybrid of those two approaches.
Can I pick up on one of the questions that's come in? It's been very interesting over the series of workshops we've had this, and this is about the limit of detection. Because Hati Saban is asking for an NGS assay. What limit of detection do you foresee being clinically relevant? How low of a variant allele frequency do you think would be a cause for change in clinical care? So we've been seeing, it's been coming down and down and down as the science gets better, but we're biologically a long way away from zero. So what do you think? Do you have a feeling for this, Chris?
Yeah, absolutely. And, the bar for us has been raised because, we're actually, we're talking about, limit of quantification and then a clinical decision point. So that's somewhat different from that. The FDA guidance to industry set us a very challenging bar, which said our clinical decision point should be a log above our technical limit. And so I think the technical capacities depend on the mutation type for NGS specifically, whether it's an indel versus a single nucleotide variant. But broadly, I think we're confidently at 1 in 1,000 limited detection of 0.1%, where we can confidently say for most regions on a panel, we can call it that level. Obviously, we can go deeper than that in the lab with greater input and greater sequencing depth, perhaps to 1 in 10,000 with technologies like duplex sequencing, for example, and even deeper still for insertion mutations like the NPM1 and the ITD mutation. So technically, I think we'll continue to push the limits. I would say the key question is really is the clinical utility piece. Is it informative to push to that limit at that particular time point? And I think we may have different LODs needed for different variant types at different points in treatment. And one of the most disheartening things for me is to spend a lot of money sequencing very deeply and then find out the mutation was actually easily discoverable at 2% or 3% in someone in remission. And so I think we may be in a situation where we have an initial standard validated panels where we can make a confident call and then reflex to even more sensitive tests in those patients who are still negative. But the current ELN guidance is 0.1%. 1 in 1,000 for NGS. That will evolve over time.
Can I ask you? Sorry, John. Please go ahead. No, just a direct question to that, because when we look at myeloma, the limit of detection is much lower. Obviously, they have a few technical advantages, but from your perspective, both technically but also clinically, what can we learn from myeloma for the application of MRD in AML?
Yeah, absolutely. So the myeloma situation is fascinating, and they really have the perfect with a for those who are not familiar with this is rearranged immune receptors, which add a perfect target for sequencing. So it's possible it's possible there's now validated tests to 1 in a million limit of detection. The concordance with other technologies seems good, at least to 1 in 100,000. down to the level we start getting into Poisson distribution issues and sampling bias, and then really the clinical utility question of what's the significance of those very low level calls. And I think we've seen some of that in our analogous variants. So FLT3 ITD, for example, where it's possible to sequence very, very deeply, but the significance of a single molecule sequenced, we're actually at the really at the limits of the technology and the just the intrinsic properties of the sequencing machines, right? There's some, even with the best approaches, there are some limitations there. So I think myeloma is clearly ahead of AML, both in the application of MRD as a clinical test, but also in the development of NGS and the pushing of NGS. So I think for many ways that makes our route, especially with the regulators, easier in that if we have some examples from NGS. But it's I think it is a different problem, and the clonal complexity in AML and the fact that many of these mutations are present in healthy older people does actually make it a different challenge to myeloma. But I think there's a lot to be learned from that field.
Thank you. John, would you pick the next question?
Unmute here. Yes, I was interested in, there's a question about duration of therapy. Georgia Campano asks, for those, sorry, it's Alan Williams asking this question, not Georgia. For those patients MRD negative and surviving for more than 5 years, were they on continuous AML treatment and frequent MRD measurements? So there's two points to that. Should you continue to do MRD measurements when people are in so-called remission? And if you get to 5 years, were they still on maintenance therapy or not?
Yeah, so the examples I showed were with a post-allogeneic transplant, and so arguably, yes, there was still a graft versus leukemia. That's a living drug we're giving, and amazingly, after all these decades, I don't have a good way to quantify anti-leukemic efficacy of a transplant. Right? I don't have a biomarker to tell how a transplant's working or a predictive biomarker of who it will work in. I can look for the mechanism of relapse. I can look for things like HLA loss in a relapse sample. But our presumption is, yes, this is essentially maintenance therapy and that there is a allo immune response throughout there. The point where we stop, we know that most relapses post an allo transplant are in the first 2 or 3 years afterwards. One of the things the measure protocol with a CRB-MTR will help us establish is we have very dense time points of sampling. And so every month for the first 6 months, and then every 3 months out through 18 months. And really the idea is are the optimally informative time points. There's nice data from Peter Hochland and Hans Ommend from decades ago showing that the kinetics of relapse differ between molecular subtypes in AML. So again, it's going to be a compromise. But I think there's a good argument for at some point saying your chance of relapse is so small, we're not going to continue to monitor you at some landmark time point that I can't define.
Yeah, from a very practical point of view, on those long-term follow-ups, are those people just having peripheral blood monitored, or most of them having marrow as well?
Yeah, so this was the data I showed there was from the single landmark from the only measurement we're using there was from pre-transplant. Okay. So this is stratifying on the basis of pre-transplant. But obviously we refine those estimates if we continue the monitoring. And I would dearly love to have blood be a sufficient sample, some component of blood, whether it's cell-free DNA or whole blood or some pre-enriched fraction of white cells. To be the sampling modality. And one of the hopes of the measure protocol is we can see if that's possible with increased sampling frequency, can we get similar resolution as doing bone marrow examination?
Certainly. Patients would obviously be very grateful for that. There's a question related to this from Larissa Higgins. For patients in the Hutch study that were MRD positive but did not relapse, have they continued to be followed up? And did most eventually relapse? Was their MRD followed over time post-transplant for those who didn't relapse?
Yeah, so that's a great question. And, these are the practice pattern at the Fred Hutch is typically myeloablation if you can tolerate it. I think there's variation in centers in the US, but they'll have a bias to doing myeloablative transplants. It's common to see MRD clearance immediately post-transplant in the recovering. And so the optimal point to measure the impact of conditioning intensity hasn't been fully refined. We have obvious time points we can look at: day 30, day 60, day 90 to 100 are all clinical landmarks we look at now. But where the optimal landmark is to measure the effect of conditioning intensity hasn't been well defined. But yes, it's entirely possible that some of those people who are MRD positive pre-transplant had an impact to the therapy. The reason we do transplant is because it works, and they may have had clearance of the MRD, and so that's entirely possible. The reason for me showing that data is because I think the Hutch is a wonderful transplant center, and they had a wonderful flow expert in Brent Wood who had really the gold standard testing for the US. And so I feel they can take it. I think the centers that, probably the test isn't as prognosis, isn't such a prognostic as the Hutch, but they're really, a leading light in flow in the US. So it's not a criticism of all of them. It's just to show how much room we have to improve in the field.
It's such a complex disease. I admire those of you who stick with it for so many years. I'm back to the days of 5% blast with a single hematologist looking down a microscope. So, you almost produced too much data. There's such a wealth of data that I can quite understand how difficult it is to fall back. Axel, I don't want to hog the conversation.
No, but I would like to pick the next question from Jacqueline Cleary. Do you feel the limit of detection and the total number of collected events important in the harmonization of flow cytometry?
Absolutely. And this is a key thing in the ELN guidance, both in the 2017 and the latest updated 2021 guidance, saying, you need a sufficiently large denominator. So you need to collect enough events to reliably state the MRD calls. One of the things we've discussed in the ELN group is should MRD assays be annotated with the proven limited detection of the assay. It's not meaningful to say an assay is negative if you're only collecting 100 events, right? So so yes, absolutely. And there's explicit best practices in the ELN guidelines of what should be done for flow.
Thank you. The next question is regarding to NGS. You said the current AML NGS assays available are not fit for purpose. What do you believe are the 3 key things that an AML NGS assay should have?
Wow. So, and when I don't want to put all the blame on the assays themselves. This is, they are designed for the purpose they're designed for. So what I'm talking about is targeted resequencing of recurrent somatic mutations, so-called myeloid panels that we often use in clinical practice of these public mutations that come up time and time again in AML. And their use case, what they were designed for, was to look at a diagnostic sample and say, what are the key mutations in that diagnostic sample? Typically, the data is reported at variant allele frequency greater than 5%. And those assays work perfectly for that use case. They're great. And there's many different companies who produce those kind of assays. They're all pretty reliable and reproducible. Where we get into trouble is when we go below that point, that 5% variant allele frequency, which those assays were never designed to do. And there's a number of reasons why they don't work reproducibly beneath that level. The intrinsic error rate of the sequencing machines we use is one assay. The fact the libraries themselves can be optimized in ways that aren't necessary for a diagnostic assay, but are for an MRD assay. So things like unique molecular indices to make sure you're not counting the same molecule multiple times, and that any introduced errors in library application or in sequencing can be corrected for. We're now at a point where we can actually put these unique molecular identifiers on both strands of the DNA, so-called duplex sequencing, which probably gets us a log more confidence in our variant calling. And so to answer your question, I think the regions of interest in an NGS AML MRD assay should be things that have known clinical utility. We can measure lots of things and they may not track with disease burden, and so it should be known. They should be, there should be appropriate specificity. And so we shouldn't be in a situation where we're wrongly assigning patients to an MRD positive state, because the implications for that are obviously more therapy, and I think that's disastrous. On that point, I'd caution people for, there's this thing of, oh, we'll just unmask the diagnostic assay and we'll look for that, we'll look for the variant and see if we see it. There's a lot of noise down in that level, which is false positive error rate. And so you want to be, want to have that high specificity, and that's something we've prioritized in my lab over sensitivity, to really say you can confidently say that's a real mutation. And then the final piece is interoperability. The test done in one lab should be the same as the test done in another lab, and one patient should be able to be referred to clinical trials. We should be able to discuss their prognosis, as a community. And if it's the whiz-bang assay that only your guy in the basement can run and nobody else has it, it's not useful. And so we really do need to come together as a profession and work on harmonizing. Even though all the incentives are to not do that, I think for patients, that's what's important, is to have a harmonized assay. So I think we just need to do that.
Brilliant. I think I know the answer to this question, but I'm going to put it anyway. So to what extent does the positive MRD reflect and is predictably based on natural level of aggressiveness and malignancy of the disease?
Great question. And so, I often, when I'm on the inpatient wards, reflect back to my clinical mentor, Professor Karp, who ran the leukemia program at Hopkins. And she had this saying, which, she taught me lots of science and medicine, but the things that stuck with me were the sayings and the anecdotes. She'd rather be cured of a high-risk AML than die of a good-risk AML. And I think, that's what we're doing. We're refining those initial prognostic classifications to give a patient-specific assessment of what's left. Absolutely correct that the poor risk AMLs are going to be more treatment refractory, more likely to be MRD positive, but there's going to be individual variation there. And so I think that's, we're all Bayesians as physicians. We like to update our pretest with new information. This is giving us iterative more information of how we're doing. Having said that, there are some, we haven't spent a lot of time investing in, for example, a complex karyotype TP53 biallelic mutated assay, for example, because the risk group of that group is just so bad, unfortunately. And so there are some groups where I think we're never in a sufficiently good remission that an MRD test would add additional information. But for many patients, I think it does offer the possibility of adding additional information beyond what's from the initial diagnostic risk grouping.
Well, I think that also addresses that last question on the Q&A function. We're nearly out of time. I'm going to hand back to Axel to say any closing remarks that you want to make, Axel, and then I'll just
one more question I would like to ask for the 4 minutes we have remaining. Thank you so much, Chris. I know it's only a brief answer possible, but how do you see the regulatory use of MRD as a patient selection and as an endpoint? One of the attendees has kindly posted the link to the FDA guidance into the chat. So just a few words, what can you leave with us on this topic?
Yeah, so I'll be careful to specify. I cannot comment on another federal agency, and I would point you to the FDA guidance. And but having said that, so I'll leave it. When you have, I believe at the last meeting you had in London, you had representatives. You had Nicole Gormley from the FDA. You had from the EMA, and so I've done really great work in helping us as a community, really challenging us with high bars. But I think the opportunity for the use of MRD goes beyond the clinical indications and really in drug development, could really speed up drug development in a number of ways. And I think the partnership we have with the pharmaceutical industry now, with things like the FNIH, really reflects that enthusiasm for using MRD in obviously as a surrogate endpoint for overall survival is the biggest goal, to really speed up trials for accelerated approval of new drugs, but also for drug development. And I think there's a stage, a stage earlier than that in assigning assigning patients into clinical trial, identifying patients at high risk. Blinatumomab in ALL would be a great example of that, where the label for blinatumomab was changed on the basis of MRD positivity being seen as an unmet need in patients in remission, with analogy to adjuvant chemotherapy for solid tumors, right? They're not sufficiently treated and are at a high risk. And then I think even earlier than that, in drug development, in early discovery, they're really impactful. Risk is a key part of that early drug discovery component, and having a better readout of the individual leukemic efficacy is information I think you would want as an industry to really know, is your compound really hitting the leukemic clone? And I'm not in that world, but I can imagine being deeply unsatisfied by having CR and just 5% cutoff being the limit of how well I can tell if my drug's working. I really want a greater range of detection than that. And so I think in early trials, I think we're increasing CMRD to get, as I said, a bright signal. If you really have many log clearance of the AML with a drug or drug combination, that may help you make decisions on whether that's a drug or combination you take forward to further trials.
Well, thank you very much indeed, Chris. It was a great question, Axel. We've run out of time. We've got to wind this meeting up, but I want to just firstly, of course, just highlight the upcoming events, and do please, all of you, keep up to date with the CDDF live webinar, and particularly you're very welcome to the workshop which will follow this excellent webinar we've had today on the 25th and 26th of April. I want to thank you, Chris, most particularly. Absolutely outstanding talk, but the discussion and all the people who've asked questions, because sitting here listening to this, I can't think of any better example of how modern science is being translated right into the heart of clinical decision making. And it's very complicated, as everybody in the field realizes, but it's very exciting. So thank you all very much for your participation. Do follow us on CDDF. And Axel, I hope you get better soon.